Uh, so we have to study oncocerciasis today. It's also parasitic disease caused by Oncocerca vulvaris, which is a parasite, a nematode actually, a nematode parasite. We also call it filaria. These thread-like parasites are also called filaria, and it's a neglected tropical disease or NTD. It's a list. The World Health Organization has identified some diseases. It's a list of about 20 different diseases uh, under the neglected tropical diseases. Okay, so why they are called neglected tropical diseases? Because not many people or the world is engaged in, doing productive research or undertaking ambitious projects in these areas, because these diseases sometimes are limited to some region of the world, like the tropical region of the world, Africa and other subtropical regions where you can find these diseases. Okay, but they are not common in other parts of the world. It's a disease of the eye and skin. Okay, and it's also called river blindness. It is caused by this parasitic worm. As I told you, this is a nematode, Oncocerca vulvaris, and it's transmitted through repeated bites by black flies of the genus Simolium. So this is a black fly that you saw here. This black fly is a vector of this particular parasite, uh, Oncocerca vulvaris. Okay, and the vector lives and breeds near fast-flowing streams and rivers, and the infection can result in blindness. Okay, this vector is found near the fast-flowing streams in the tropical region, near the rivers, and it can bite people because it sucks blood, which this particular vector needs for ovulation. And along with that, it also passes on this particular parasite or the nematode to the people. So this skin disease that is causing is in the form of these nodules that you would see on the surface of the skin or under the skin, and it would cause a lot of itching as well when you are infected. And the vector, it's a day biting vector, so it would bite people during the daytime. There is no vaccine available for this particular disease. If you look at the distribution, this is the map which uh, was generated by WHO a while ago, where you can see these different countries where this particular problem is prevalent. So most of the countries you can see are African countries, and some of the countries in South America also have this problem. So it is prevalent in both these parts of the world, in Africa and South America. Um, and you can see the geographical locations are different, but the conditions, if you look at the tropical or subtropical locations of the countries, it's more or less the same, because the tropical line would run here of the world, and this some of the countries are near the tropical region and other are subtropical region. So these are the areas where this particular problem is prevalent. These are some common related terms. River blindness, we call it the river blindness, or let's say, uh, if you turn off your mic, then we can, there will be no disturbance. If somebody's mic is on, they can. Okay. Yeah, that's right. Okay. These are the common related terms: the river blindness, African river blindness, blinding filariasis, or Robles disease, croc. So these are different terms used for the same condition. And some other clinical terms you can see here: the oncodermatitis, oncocercical dermatitis, lipid skin. So you can see these are different terminologies that you can go. They are essentially describing the same condition that we are talking about here. And I told you about the causative agent, which is this filarial parasite Oncocerca volvulus. Main species found in the skin and tissues, okay? And the microfilaria of O. volvulus are unsheathed with broad spatulate head and pointed tail free from nuclei. That is the physical appearance of this particular parasite when it is under the skin. They measure between 221 to 287 micrometer long and migrate to the dermis, causing itching and skin texture changes, and occasionally arrive in the eye where they cause blindness. Okay, so they can travel under the skin to different parts of your body, and that's they can along the way they can cause this kind of itching and these problems, because I have told you about the immunology of parasitic infections, where you can see that these different type of mast cells and the basophils and eosinophils, these actually are providing immunity against the parasites, and their degranulation can cause different kind of symptoms or different problems, including itching and this redness of the skin area where they encounter some kind of parasites. The vector is the black fly. It belongs to this family, Simulidae, and black flies, also known as buffalo gnats, they are small flies, 1.5 to 4 millimeter in length, and normally black in color with short hairless legs and antennae. And they have large compound eyes and characteristically hairy humped thorax, and can give a painful bite. Black flies slash the skin and lap up the pooled blood. Okay, the saliva injected uh, while feeding causes swelling and soreness that can persist for days when it is going to. So this is the filaria, uh, the black fly that we are talking about. Okay, so the oncocercical infections are found in tropical climates. The main burden is in the sub-Saharan Africa, though the parasite is found in limited areas in the America and even in the Middle East. That's what I told you. And that is the burden of infection worldwide. People who live near streams or rivers are at higher risk of contracting the disease, and you need several bites for the infection to occur. If you look at the life cycle of this particular uh, parasite, that how does it complete its life cycle in two different hosts? One is the black fly and the other is human, because they will live closely in these different parts of the world that we are talking about. So the female black flies need to ingest blood for ovulation, so they feed on humans. And that's what I told you earlier, that these flies need blood, and they would suck blood, from the human being or other animal species. Larva can be ingested by the black fly after biting. Okay, when it bites a particular person, then the larva uh, can be ingested in that particular uh, during that particular biting. It becomes infective for humans in about 10 to 12 days. So it would take some time for the that particular larvae which are there in the saliva of the fly to become infective, and that's the range is from 10 to 12 days. Once inside the human body, the larvae mature into adults in around 3 months to 1 year. Again, you can see that it's taking a lot of time. It's a broader range from 3 months to 1 year. Uh, this larva will mature inside the human body. Most adults worms live in fibrous nodules. As adults, female worms produce thousands of new larvae daily. So most of these these worms that are maturing, they live there in the fibrous nodules under your skin. In this lymphatic systems, uh, which is running alongside your blood vessels. Okay, so it is living in these nodules there, and it will live there for a long time. The adult worm can live up to 15 years inside the human body or inside these nodules. And during this period, these worms, uh, worms can be produced by a female for as long as 9 years. So it can be uh, it will be reproducing for 9 years, producing new types of worms in your body. The infections can be asymptomatic, or symptoms include these itchy skin, nodules that we talked about on the skins, and vision changes. If it infects your eye, there would be vision changes. And there can be non-painful swelling of lymph glands, but this is not common. The symptoms are usually caused by immune response to dead or dying larvae. When the larvae are dying, then they would come in contact with these different type of immune cells that we have learned about during our lectures on immunology, and those cells would release different kind of substances which would produce these symptoms that people would feel. The inflammation caused by larvae that die in the eye results initially in lesions on the cornea, but without treatment progress to permanent clouding of the cornea resulting in blindness. Okay, so uh, these parasites, when uh, when this particular fly injects the saliva into the skin of this particular human being, so it would just live there for some time in the subcut tissue or subcutaneous tissue, and then the adult swarm would arise in the subcut nodules. It would take uh, a lot of time. As I talked to you, that it can take about 3 months to 1 year time, and then the adults produce these unsheathed microfilaria that typically are found in the skin and lymphatics of connective tissues, but also occasionally in peripheral blood, urine, and sputum. So there are other tissues that you can find, or other body fluids where you can find this microfilaria after you are infected. Okay, not only these sites, the lymphatics and the skin tissue that we are talking about. So these adults would produce these unsheathed microfilaria under the skin, and they can be found in other tissues as well. When the black flies, they would take another blood meal. These microfilaria, the unsheathed microfilaria, uh, are already there inside the blood. So the black fly um, would just take a blood meal, and along with that, it would also take these microfilaria. There are different stages of microfilaria uh, inside this black fly. When the black fly ingests on this larvae, then the larvae would undergo uh, different changes. Okay, there are different stages: larva stage 1, 2, 3. Okay, and when it reaches to stage 3, then it migrates to you know uh, these migrate to head and the proboscis of this particular fly, and from there it is now ready for another cycle. So it is the third stage larva actually, which would be uh, injected into the human skin. From stage 1 to stage 3, this development of the larva is taking place inside the fly, uh, this black fly that we are talking about. And the third, from from stage 3 onwards, you can see that it is an infective stage for the human being, and uh, it would start the human cycle after injection. And these are different pictures of people who are infected with this particular parasite. You can see the lesions here. These are the lesions on the skin. You can see these small nodules. You can see these bigger nodules under the skin. These are also caused by this particular parasite. Or you can see these lesions on the foot or the legs. You can see here it is on the forehead. So some these skin lesions or these eye problems are caused by this microfilaria, and it travels to for different parts of your body under the skin. Okay, so it is long lasting. It lives there for a long time, and it migrates slowly to different parts of the body and cause these different problems that we are seeing here. How do you diagnose it? The golden, the golden approach is the skin snips. Okay, it is the gold standard. We call it the gold standard because it's the best way to diagnose it, and it can be available to many people as well. Okay, so what is a skin snip? So you just you can look at this picture. You can just flip a piece of a skin from some area of your body, and then you can cut it with a scalpel, and then you can put this skin tissue in normal saline for 24 hours, and the microfilaria, if there, they would come out in this in a matter of 24 hours. And after that, you can examine that tissue under the microscope and see these microfilaria. You can also go for other approaches to use the skin for DNA extraction and PCR. You can also do PCR. Okay, PCR can be employed for this purpose. If you have extracted DNA from the skin, or if you have blood sample of a person who is infected, so in that case also you can detect these parasites with the help of uh, this particular PCR. Then the second most popular test which is used is the Masotti test, and the Masotti test is you can say can you see this slide?
Yes, sir.
Yes, sir.
Uh,
Sir.
can you see this Masotti test here? No. The description of the Masotti state, it's a website actually. Maybe I have not shared it with you. It's okay. I don't know how to share, but I would just relate it as it is written. The Masotti test, you know, if the skin snip is negative due to some reasons, if you couldn't figure out these parasites under the microscope, then you take some oral dose of DEC. Okay, it's diethyl carbamazine. So you take a 5 milligram dose of DEC, and the DEC inhibits neuromuscular transmission in nematodes. Okay, so it's an inhibitor of this neuromuscular transmission of the parasite. If the patient has oncocerciasis, intense pruritus results within 2 hours due to dying microfilaria. As I told you that the microfilaria, when they are dying, they would cause this itching and other problems. Okay, so when you inject DEC, this particular substance into the skin of a person who would have microfilaria, then it would cause intense pruritus. Pruritus is intense itching of the skin that would just ensue after that. And that is also an indication that this particular person is infected. But the problem with this test is that um, it can also cause some serious systemic infection in people who are not infected. Okay, so the situation can sometimes be very serious. It can be some kind of allergic reaction and some hypersensitivity reactions in people who are not infected. So that Masotti test is only applied in people who are very much suspected to have this condition or the disease. Okay. Then there is a DEC patch test. Again, the same kind of chemical is used in it, and it is applied locally. It's a local application of 10% DEC, which is applied to the skin, and it is covered with a dressing. The page is then checked for local dermatitis caused by the dying microfilaria. Okay, again, it's the same thing that you apply this page or 10% DEC, and then you remove that page and examine the skin under that particular page and see if there is any inflammatory signs of inflammation under the page or not. Okay, so this is safer as compared to Masotti, because in Masotti test you were just administering oral DEC. This is not oral, this is topical or local application of this particular uh, you know chemical, the DEC, diethyl carbamazine. There are other approaches in which you can just go for excision of the nodules, where you can possibly find out these parasites. Okay, the nodules may be present on different parts of your body, so you can just go for excision of parts of the nodules and examining them uh, via different methods. And there are ELISA tests, antibody tests. The ELISA can detect these antigens uh, inside the blood tissue. And the PCR can be used for the blood or other tissue extracts, so DNA extracted from the blood or other tissues. That is about the diagnosis of this particular condition, that how can you diagnose the condition. And the treatment, ivermectin, is recommended as a treatment because it can kill these larvae, and it's quite effective. Okay, there are also other treatments which include these doxycycline that kills the adult worms. So the different drugs are killing different stages in the life cycle of this particular parasite, and different combinations are used. Prevention and control, that is the best way. Okay, so this was about the oncocerciasis that uh, is causing river blindness in different parts of Africa. There are about 31 countries in Africa which have these problems. You saw the figures that it has crossed 25 million people that are affected by this condition, uh, you know, worldwide. On yearly basis, there are lots of people who are suffering. That's why it was the disease has been listed on the neglected tropical diseases list, which is a list of diseases that the World Health Organization is trying to get rid of by the year 2020. But 2020, you're seeing that we are facing now a different situation. That's why it would affect these eradication projects for many other types of disease, including these neglected tropical diseases. Okay, because of this corona situation that we have right now, it definitely would affect other conditions as well. Okay, so